Academic, the Registrar, the Bursar, University Librarian, Provost of the College of Medicine, Provost of the Postgraduate College, the Dean of the Faculty of Basic Medical Sciences, Deans of other faculties and of students, Directors of institutes and centers, Heads of departments, the 12th Vice Chancellor of this university, distinguished guests, ladies and gentlemen. The 609th University of Ibadan inaugural lecture will be delivered this evening by Professor Owoeye of the Department of Anatomy on behalf of the Faculty of Basic Medical Sciences. Professor Owoeye was born in Ado-Ekiti on 24 December 1956 as the first child of the family of late Mr. Elijah Adetumola and Mrs. Lydia Bosedi Owoeye of Ikogosi Ekiti. He attended Oke-Ibadan High School Ibadan in 1972, where he gained double promotion from Form 1 to Form 3. He thereafter completed his secondary education at Ifeoluwa Grammar School, Osogbo, where he graduated with the best West African School Certificate results of Division 1 distinction. Division 1 distinction with aggregate 12 in June 1975. He proceeded to Government College, Ibadan, for his advanced level studies between 1976 and 1978, and was admitted thereafter to study medicine in 1978 at the University of Ibadan. He completed his preclinical and clinical training at the College of Medicine in June 1983, earning the Bachelors of Medicine and Bachelors of Surgery, bagging 2 awards. First is the Latude Odeku Prize in Neurosurgery and the Glasgow Prize in Pediatrics. He served the mandatory 1-year National Youth Service Corps program NYSC in Kano State and did his primary assignment at the Psychiatric Hospital Goronduse, Kano, between 1984 and 1985. Thereafter worked briefly at the as a medical officer at the Castle Clinic, No Man's Land, Kano, and later as a medical officer at the Department of Surgery, Murtala Mohammed Hospital, until late 1987 when he returned to Ibadan. He later worked at May Clinic in Alende and Alafia Tayo Hospital in Ibadan, before proceeding to pursue a master's degree in anatomy in 1999 at the University of Ibadan. Professor Owoeye joined the services of the University of Ibadan on June 8, 2001 as lecturer grade 2. He was promoted to lecturer grade 1 in 2006, and on completing his PhD in 2010 was appointed senior lecturer in 2011. He was promoted to the grade of reader in October 1, 2015 and professor on October 1, 2018. He was a visiting scientist to the Hussein Ibrahim Jamal Research Institute for Chemistry, International Center for Chemical and Biological Sciences at the University of Karachi in Pakistan, and the host of Professor Iqbal Chowdhury between 2006 and 2007. Professor Owoeye has served the University of Ibadan in various capacities in the last 25 years. This includes departmental coordinator for postgraduate programs between 2010 and 2015, coordinator for MBBS programs between 2007 and 2010, and teacher for gross anatomy between 2007 and 2010, officer in charge of embalming facilities between 2002 and 2015, officer in charge of anatomy specimen museum between 2002 and 2015, acting head of department of anatomy between 2018 and 2020, acting head of department of anatomy 2022 to 2024, and substantive head of department between 2024 till date. Professor Owoeye was a member technical committee on teaching aids in the College of Medicine in 2004, member college counseling committee in 2010, Secretary General, Ibadan College of Medicine Alumni Association Worldwide between 2011 and 2015, member of the Academic Board of the College of Medicine between 2014 and 2016, member College Research and Innovation Management CREAM Unit between 2017 till date. He is a member of the University of Ibadan Senate between 2011 till date. Warning: Alexander Brown Hall, University of Ibadan, Between 2012 to 2013, Assistant Hall Warden, Alexander Brown Hall, University of Ibadan, between 2007 to 2012. Professor Olatunde Owoeye was External Examiner and Assessor to the Department of Anatomy, Ladoke Akintola University, Lautech Ogbomoso. Between 2012 till date, Department of Anatomy, Bowen University, Iwo. Between 2014 and 2015, Department of Anatomy and Cell Biology, Obafemi Awolowo University, Ile-Ife. Between 2016 and 2020, School of Laboratory Medicine and Medical School, College of Health Sciences at the University of KwaZulu-Natal in South Africa. Between 2017 till date, Department of Anatomy, College of Health Sciences, Olabisi Onabanjo University at Owoeye, 2021. Department of Anatomy, College of Health Sciences, University of Medical Sciences, Ondo, between 2021 to date. Department of Anatomy, College of Health Sciences, University of Ilorin, between 2023 to date. Department of Anatomy, College of Health Sciences, Ahmadu Bello University, Zaria, between 2023 to date. Department of Anatomy, Medical University, Lagos. Department of Anatomy, Lagos State University College of Medicine, Ikeja, between 2023 till date. Department of Anatomy, University of Lagos, Akoka, between 2023 till date. And the Department of Anatomy, Babcock University, Ilisho, Remo, between 2024 to date. Professor Olatunde Owoeye, as a distinguished academic, have reviewed quite a number of journals, some of which include the International Journal of Biological and Chemical Sciences of South Africa, Tropical Veterinarian, University of Ibadan, African Journal of Medicine and Medical Sciences, UIUCH, University of Ibadan, Nigerian Journal of Physiological Sciences, University of Ibadan, Nigerian Veterinary Journal, Journal of Nigerian Veterinary Medical Association, Journal of Dietary Supplements of the United States, Archives of Basic and Applied Medicine, College of Medicine, University of Ibadan, and the Nigerian Journal of Neuroscience. Professor Owoeye is a member of the following learned societies: the Nigerian Medical Association, American Association of Anatomists, Anatomical Society of West Africa, of which he is a fellow, Anatomical Society of Nigeria, Society of Experimental and Clinical Anatomists of Nigeria, Society of African Neuroscientists, the Nigerian Society of Neuroscientists, and the Association of Specialist Medical Doctors in Academics. His research interest is in neuroanatomy with special focus on neuroprotection, and specifically in the structural modifications that accompany neurotrauma and neural regeneration using drugs and plant medicinal products with antioxidant activities and observing their neural responses. A seasoned academician and researcher, Professor Owoeye had successfully supervised 4 PhD theses, 2 ongoing PhDs, 26 MSc dissertations, and 3 BSc projects. He has over 85 publications published in reputable journals to his credit, including a chapter in book, journal articles, and conference papers. His Google Scholar citation count is 1,954 with an H index of 22. Professor Owoeye received a letter of commendation from the 11th Provost of the College, Professor Oluwumi Olapade Olaopa, in 2020, and has received many recognitions from the University of Ibadan Medical Students Association many times as the best lecturer in preclinicals as the best lecturer in preclinicals 2009, 2010, 2012, Face of Preclinicals 2023 and 2024. He has also received Merit Award from the Alexander Brown Hall as awarded in 2013. Professor Olatunji Owoeye is a member and national officer of the Full Gospel Businessmen's Fellowship International, Nigeria, between 1994 till date. He is also a member of the Special Marshal Unit of the Federal Road Safety Commission between 1992 and 1995. President, Ifeluwa Grammar School Old Student Association, Oshogo, between 1992 and 1995. Secretary, Police Community Relations Committee, Ogbere Police Division, Ibadan, between 1993 and 1995. Member, Faith Academy Governing Council, Badeku, Ibadan, between 2011 and 2013. Professor Owoeye is happily married to Fumilayo Elizabeth, proprietress of Higher Heights College, Olomi, Ibadan. And the marriage is blessed with 3 promising children: Barrister Oluwagbemiga Owoeye, Mrs. Iyanoluwa Alawode, and Faith Owoeye. He is also blessed with 4 grandchildren. Distinguished ladies and gentlemen, I now invite Professor Olatunji Owoeye to deliver his lecture, which is titled The Human Brain, the 3S, THEREIN, and Neuroprotection. Professor Owoeye.
The Vice Chancellor, sir. Deputy Vice Chancellor Administration, Deputy Vice Chancellor Academic, Deputy Vice Chancellor Research, Innovation, and Strategic Partnerships, Registrar, Bursar, University Librarian and of the Postgraduate College, Dean of the Faculty of Basic Medical Sciences, Dean of other faculties and of students, Directors of institutes and centers, distinguished ladies and gentlemen. I give God all the glory for this opportunity and honor to stand before this distinguished audience in this hallowed chamber today to present the 609th inaugural lecture on behalf of the Faculty of Basic Medical Sciences, College of Medicine, University of Ibadan. The first, the best, and the preferred. It is indeed a great privilege to be asked to give this lecture after being promoted full professor of anatomy 2018 inaugural lectures. Adebowale 2020 gave a detailed and lucid insight into what inaugural lectures of the University of Ibadan constitute, namely a central part of the university's academic tradition, events here to commemorate appointments, a platform for an academic to present the body of his research, to provide the university the opportunity of showcasing its academics, to present a call for celebration and reflection, to signal the beginning of further and higher intellectual achievement, and a formal speech by a new professor. The first inaugural lecture. This inaugural lecture is the first from the Department of Anatomy, and if I'm standing before you, it is because of God's mercy and the men that I met on my journey in this university. The Department of Anatomy has had many giants. Professor Alastair G. Smith was the first head of the department. Professor T.I. Grillo was the first Nigerian head of the department. Professor A.B.O. Desalu, a fellow of Anatomical Society of Nigeria, and the only life fellow of the Anatomical Society of Nigeria, was the next day Professor N.T. Shokunbi, the anatomist neurosurgeon, who later became the chief provost of the College of Medicine. He was the one who reactivated and recommenced the postgraduate program in anatomy. A beneficiary of that program is the inaugural lecturer of today. We were followed by Canon A. Malamo, the anatomist neurosurgeon, who was head of department and later dean, Faculty of Clinical Sciences. He was 3-in-1 to me. He was my HOD, he was my PG lecturer, and also was the supervisor of my research project. The list of our previous HODs is displayed. I have mentioned the first 4, and we can read through the list. We move on to postgraduate studies in anatomy. Postgraduate studies was not alien to our department, as the bookshelf in the head of department's office boasts of evidences of active postgraduate experimental research with the PhD thesis of the following lying on the shelf. Dr. A.B. Odisal 1973, Dr. A.B. Otola 1977, Dr. P. Ogunake 1979, Dr. P.V.V. Prasada Rao 1981. But today the story of the department has changed dramatically by innovative leadership, dint of hard work, resilience, determination, and prudent management of resources. Apart from 4 functional research units, the department is equipped with postgraduate programs that now attract Africans from every corner of this country for our PhD and MD program. Mr. Vice Chancellor, sir, my journey to lectureship followed this pattern. I came from A-level 1976-1978 class of Government College, admitted October 1978 to the MBBS class, and then graduated June 1983. Then I went away for my housemanship, NYSC, work in Kano, work in Ibadan. I became born again December 1993 at the Living Spring Church, and then I joined the Full Gospel Business Fellowship in 1994. In 1998, the Lord spoke to me, go back to academics, and I obeyed. So I returned. And started the master's program in anatomy as a member of TRIO. The other 2 people were Echo Kobe from Lautech and Security Timo from Ilorin. Then I came back and accepted. I had an opportunity to be given employment in the Department of Anatomy, so that by 2001 January, I was a lecturer grade 2. Mr. Vice Chancellor, sir, I'd like to briefly review what anatomy is all about. It's not only about corpses and morgues, no. Lecturers have told me anytime I say from anatomy, they say, is that the place we bring the dead body? I said, yes. Well, we have more than the dead bodies. The word anatomy derives from the Greek word anantenein, which means to dissect or to cut apart. It's actually the scientific study of the morphology of the human body. The early anatomists like Aristotle, Eratosthenes, Abe, and co. laid the foundation from knowledge that they obtained initially by dissecting animals and later human beings. At that time, anatomy was simply speculative or merely descriptive, but anatomy now is a central science that is numeric, involving measurements, data acquisition, data storage, statistical analysis, and experimental. What are the divisions of anatomy? There are 4 major divisions, namely gross anatomy, microscopic anatomy, developmental anatomy, which we call embryology, and neuroanatomy. The composition of the human body, as complex as it is, consists of cells, which is the smallest unit of the body, that form tissues. The tissues form organs, the organs form systems, and it's the system that form the organism. There are 4 basic tissues in the body, namely epithelia, connective tissue, muscle, and nervous tissue. What is the division of the nervous system? It consists of the central nervous system, the peripheral nervous system, and autonomic nervous system. We now look at the composition of the nervous system. It consists mainly of the most fundamental of the cells called neurons. Neurons are the functional unit of the nervous system. Synapses are the sites of functional contacts with other neurons or with other glands. Neuroglia is the packing tissue, and there's a covering for the brain and the spinal cord, which we call meninges. Why is my interest in the brain among the other structures in the body? Mr. Vice Chancellor, sir, my interest is that the brain is the chief executive officer of the human body, where major decisions concerning life are taken. I chose to love the brain since 2000, mainly due to the fact that that was the organ that my PG teacher and MSc supervisor, then Canon, Dr. Adefolari, Obanishola Malomo, the anatomist neurosurgeon law. I was applying what I read on page 62 of Professor K. Radimowale's Inaugural. He said, I am reminding you of two things. The apple does not fall from his tree, and a good mentee will also reflect his mentor. So if he was dissecting the brain, I also was dissecting the brain. While my mentor was busy cutting and dissecting human brains, I also have been busy in the last 25 years cutting and dissecting rat brains. After all, brain is brain. Okay? But there's a difference. The difference is that he does the dissections of the human brain to save human life from life-threatening disease. Whereas in my own case, my subjects are rats. So when his subjects wake up and they will thank him for a job done, my own subjects don't wake up because they will sleep no more. It is not that I am wicked. No, it is because I am trying to gather data that will contribute to knowledge. I hope Mr. Ras, you understand this point. What is the importance of the human brain? The human brain consists of two cerebral hemispheres joined together by a mass of white fluid called corpus callosum. The human brain weighs just about 1.4 gram, which the human brain is the organ that is solely concerned with intellectual function, thought, memory, consciousness, attention, emotional experiences, ability to sleep, creative ability, decision-making, complex perception, judgment, planning, orientation, speech, and language. Only Homo sapiens can speak and act language. The capacity for speech and language is unique among all, for Homo sapiens, among all other animals. Mr. Vice Chancellor, sir, my inaugural lecture is titled The Human Brain. The three is therein and neuroprotection. It has two parts. The first part is speaking of my observation as I read, studied and noted the occurrence of many threes in the various parts of the central nervous system. The second part will consist of my contribution to knowledge via the researches I have conducted all the years you have employed me in the University of Ghana. The threes. The neuron is the basic functional unit of the nervous system. Neurons may fundamentally be classified into three morphological types, namely the unipolar, the bipolar, and the multipolar. The brain and the spinal cord are delicate semi-solid structures that need protection and support. Both structures are encased in a bony vault, floated in cerebrospinal fluid, and invested by three membranes. These three membranes that invest the brain and spinal cord are called the dura mater or Pachymenes, the arachnoid matter, and the pia mater. Each peripheral nerve is composed of one or more bundles called fascicles of nerve fibers. The three protective structures that ensure the structural integrity of a peripheral nerve as it traverses the body are named perineuron, endoneuron, and epineuron, the spinal cord. Our ability to stand up from where we were and to walk into this room this afternoon, our ability to stand, to walk, to run, to dance, and play football are all dependent on the spinal cord. The three horns of the gray matter of the thoracic part of the spinal cord consist of the dorsal horn, the intermediate horn, and the ventral horn, each of them having specific functions. The brainstem. The three components of the brainstem are the midbrain, where visual and auditory reflexes reside, the pons, and then the medulla oblongata. This is the center that contains all the control for consciousness, for swallowing, for breathing, and digestion. Mr. Vice Chancellor, the cerebellum controls our ability to turn the key to open our car, to open our office or our office. The cerebellum also controls our ability to turn the ignition of our car, to stand and maintain our balance on the spot, to maintain a steady hand when we carry hot tea or cold water from a table and we bring it into our mouth. The fact that it does not pour on our dress is because of the cerebellum. The cerebellum can be divided into three sagittal zones of cortex, namely the vagal zone, the medial or paraverval zone, and the lateral zone. Purkinje cells. Still talking about cerebellum. Purkinje cells are highly differentiated flat-shaped neurons forming a monolayer sandwiched between the granular and molecular layers of the cerebellum. In fact, the cerebellum was named after Professor Johannes Evangelista von Purkinje, a Bohemian priest who was actually a professor of physiology. So it was a professor of physiology that named and worked on the Purkinje cells. Purkinje cell is important because it is the sole neuron that leaves the cerebellum and connects to other parts of the body. The axons terminate in 3 different ways. It could synapse on the deep cerebral nuclei, it could bypass the deep cerebral nuclei, it could go outside the cerebellum, or sometimes it go back to the upper layer or the molecular layer to synapse. The diencephalon is called the thalamus. The thalamus has 3 parts, namely the anterior part, the medial part, and the lateral part. The thalamus is important because all consciousness, all sensations that come to the body are integrated there. The limbic system is responsible for emotions. There are 3 authors, 3 researchers that laid the foundation for it, for all the knowledge we have about the limbic system, namely Willis, 1664, Broca, 1878, and Papez, 1937. The fornix, which carries all the memory fibers to different parts of the brain, has 3 parts: the body, the cross, and the column, the basal nuclei. The basal nuclei is a collection of masses of gray matter that are situated within the cerebral hemisphere. It has 3 parts, namely the putamen, globus pallidus, and claustrum. The striatum, which is also part of the basal ganglia, is the largest and most complex of the basal ganglia structure. It has 3 major subdivisions, namely caudate nucleus, the putamen, and nucleus accumbens. Mr. Vice Chancellor, sir, our ability to speak clearly as I'm speaking to you depends on the particular part of the brain. The cerebral hemisphere consists of convoluted cortex of gray matter overlying the central medullary mass of white matter. But the lobes of the cerebrum are named. There's a lobe we call the frontal lobe. That lobe has 3 gyri, namely the superior frontal gyrus, the middle frontal gyrus, and inferior frontal gyrus. It is in the left inferior frontal gyrus, which we call the Brodmann's area or Broca's area, that our ability to speak lies. Any major trauma to that part leads to slow, labored, and difficult speech. Again, there's another lobe of the brain we call the temporal lobe. It consists of 3 gyri, namely the superior temporal gyrus, the middle temporal gyrus, and the inferior temporal gyrus. It is the left posterior part of the superior temporal gyrus that our ability to make meaning and understanding of the written words or what we are reading lies. It is called the Wernicke's area. Injury to that part leads to someone talking a lot of words but making no meaning. We call it Wernicke is wordy. Cranial nerves. The movement of the eyeball to follow objects and move from one part to another part is dependent on 3 of the 12 cranial nerves, namely oculomotor nerve, trochlear nerve, and abducens nerve. My contributions to knowledge. Mr. Vice Chancellor, sir, having reported some of my findings of careful study of trees in the brain, I seek your permission to proceed now to report some of the many researches I carried out with other distinguished scholars within and outside my department and with colleagues outside this country. My research focus is in neuroprotection. Neuroprotection can be defined as a relative preservation of neuronal structure and or function. My emphasis has been to prevent or ameliorate damage to the structure of the neuron or the central nervous system, and how to repair them using chemical compounds and plant medicinal products that has antioxidant activity. Neurotrauma can arise from different areas. People can fall down, there can be injury, there could be radiation injury, chemical toxicity, if we are strips of temperature. But it has been found that something is common to them. Oxidative stress due to generation of excessive reactive oxygen, which we call free radicals, can overwhelm the body's antioxidant system, thereby linking to pathology. What has been my focus? To investigate and identify substances that will protect the nervous system from degeneration, from death, and from injury. thus limiting the microanatomical alterations and neuronal dysfunction, and thereby maintaining cellular integrity of the brain and the spinal cord. Radiation and the spinal cord. Mr. Vice Chancellor, sir, my first research interest, stimulated and directed by my supervisor, then Dr. Kano Maloma, was in the area of radiation and spinal cord. The machine we use for inducing to induce gamma radiation is found only in usage in the Department of Radiotherapy, and the machine will be shown to us in the lab before we leave. We know that exposure of biological materials to gamma radiation may impair the health of the tissue. Gamma radiation will kill cancer cells. At the same time, it will also kill the neurons or nerves or tissue that is adjacent to it. So in that first research, the purpose was to determine the possible protective effects of dexamethasone, ascorbic acid, and metronidazole or gamma radiation in spinal cord. We irradiated some rats with 2.5 grade of cobalt obtained from an AACL machine. And then we noted after we did microscopy that the smooth muscle in the anterior cells were separated, the neurons were degenerated, and the diameter of the neurons in the anterior part of the spinal cord was grossly reduced by radiation by 170%. But the treatment with dexamethasone and metronidazole partially protected the spinal cord from the radiation injury. The result of this study was what I presented in the first conference that I ever attended, the Anatomical Society of West Africa, University of Ghana, Kolebu, in 2003, and it was later published in the African Journal, Medicine and Medical Sciences. Mr. Vice Chancellor, sir, having observed the effect of gamma radiation on the spinal cord, we then proceeded to study its effect on cerebellum. We irradiated some rats with 5 gray units, 1 day old rats, irradiated them with 1 gray unit, 1 hour before irradiation. The result was we noted that irradiation significantly reduced the external granular layer, the molecular layer, granular layer, and the Purkinje cell layer, and distorted the Purkinje cell monolayer in the cerebellum. But we noted that the rats that we gave dexamethasone, these were protected. The result says that dexamethasone was protective of the damage that gamma ray was doing. The results were published in the International Journal of Morphology, 2006. Mr. Vice Chancellor, the first product of my PhD effort was the isolation of a new chemical compound from the leaf extract of Vannonia midalina, which we call bitter leaf. Having started my PhD, I had the good fortune of having the late Professor Silas Kaluonguka of the Department of Veterinary Medicine and the erudite scholar, Professor Olatunde Farombi, as my co-supervisor. With the tremendous help and support morally and financially I received from the Dean of the Basic Medical Sciences, the Professor Olaleye, and the Provost, Professor A. Migoro, I proceeded in 2006 to the University of Karachi. The many functions, folklore function of bitter leaf are listed. What was the purpose of research? To characterize the active compounds in the leaves, because my supervisors insisted we should not just work on an extract. We must know what is the active compound that is in these leaves. So packaging 4.5 kilograms of dry and covered leaves of Vannonia midalina, we fractionated after extracting to get the VAD, that is dichromethane fraction, petroleum metal fraction, ethyl acetate fraction, and the butanolic fraction. With the dichromethane fraction, it was subject to further fractionation, and a white solid was obtained, which after various spectrometer, optical rotation, HBMS, and so many other studies, we were able to isolate a compound which was resolved to contain 20 carbons, resolved into 1 methyl, 7 methylene, 5 methyl, and 7 quaternary carbons. A comparison of the spectral data of that compound showed that it was called, it was a sesquiterpene lactone, which we named epivanodalol. The implication was that we have identified epivanodalol. What is this useful for? So we proceeded to do an anti-cancer assay for this, and we found that that cytotoxic activity against skin melanoma HT144 cell line. In summary, my first research showed the isolation of epivanolol and the fact that it has anti-cancer activity against skin cancer. Mr. Vice Chancellor, sir, the radioprotective effect of metallic extract was the next work we did, and we noted that the metallic extract of Vanilla vinalina had antioxidant activity. So we tested it in Wister rats, comparing it with the effect of vitamin E, which we name which is alpha-tocopherol. Well, they were all irradiated with radiation using just 2.5 gray. And what was the result we got? Gamma radiation caused a statistically significant reduction in the means of the molecular layer of the rats, the cerebellum of the rats, the Purkinje layer, granular layer, even the widest diameter of the neurons were diminished. Whereas the ones we gave the ethanolic methanolic extract and vitamin E elicited a significant elevation of the means of the above. The implication of that is that treatment with the bitter leaf and with vitamin E protected the neurons. The results were published in European Journal of Anatomy, Owoeye et al., 2010. Mr. Vice Chancellor, sir, we then looked at the effect of radiation and protective effect that ethanolic extract of Laurine tarasosfolia can give. We noted that radiation regimes are designed to maximize tumoral side effects while causing minimal damage to normal organs. But as I've pointed out, it still will damage the neuron or the body tissue that are so close to the cancer cells. So the purpose of our research was to see if the ethanolic extract of Laurine tarasosfolia, which Yoruba people call Iyanrin, with its antioxidant activity, can prevent the brain from radiation injury. We radiated some rats, and we also gave them the extract. The result we got was that the radiation induced histological alterations in the cerebellum, in the dentate gyrus and cornu ammonis of the hippocampus, and distorted the layer structure of cornu ammonis in the hippocampus. The pretreatment with ethanolic extract of Laurine tarasosfolia significantly reduced this effect. The implication of this is that urine, which we call Laurine tarasosfolia, can prevent and did prevent radiation injury. The result was published in the Archives of Basic and Applied Medicine, Owoeye and Malomo 2015. Mr. Vice Chancellor, sir, the drugs we use in treating cancer can also cause problems for the patient. The drugs, a very important drug which is called cisplatin, is very potent and used in the treatment of cancer. But we noted that it also has neurotoxicity. We read that it has neurotoxicity associated with it. So we treated a group of rats with that cisplatin, giving only one dose. And then we gave them THEREIN, which is a free intramolecular intracellular amino acid that can cross the blood-brain barrier. And what findings did we have? The rats that were treated with cisplatin had reduced brain antioxidant defense, the elevated lipid peroxidation, elevated nitric, and then the diameter of the somata of the pyramidal neurons were reduced. Whereas THEREIN pre-treatment reversed all these alterations. The implication is that THEREIN protected rat brain from cisplatin toxicity. The result of the experiment was published in Biomedicine and Pharmacotherapy, Owoeye et al., 2018. Environmental chemicals can also injure the brain. The toxicity of mercury chloride on the brain and the modulatory effect of Lycophyllum esculentum, tomato, was another research that my project undertook. Mercury is a hazardous environmental toxicant and industrial pollutant, which on exposure causes a wide range of adverse effects. Unfortunately, mercury is easily available because it is found in batteries, fluorescent lights, thermometers, thermostats, sphygmomanometers, clinical thermometers, and even vaccines. We exposed some rats to mercury chloride, and we also gave another group of rats tomato, Lycophyllum esculentum. We noted on microscopy that the mercury chloride induced alterations in the cerebral cortex In the common harmonies of hippocampus and cerebellum of rats, the neurons were pycnosed, they had cariolysis, they had eosinophilia, whereas the one we treated with tomato demonstrated ameliorative activity that reduced the activity of the effect of mercury chloride. The implication, therefore, is that tomato ameliorated and protected against mercury chloride alteration in the microanatomy. The report of this study was published in the International Journal of Biological and Chemical Sciences, Owoeye and Farumbi, 2015. Stroke. Mr. Vice Chancellor, being a disease that is so rampant, we investigated the role of naturally occurring plants on stroke. We studied the effect of carica papaya, pawpaw leaves, on stroke. What is stroke? Stroke is the rapid loss of brain functions due to blood supply disturbance and ischemia that is caused may lead to the death of the neurons. We then gave a group of rats. We induced stroke by bilateral carotid common carotid occlusion in a group of rats, and another group we also induced stroke, but we gave them carica papaya, which is pawpaw leaves. By the time we examined them on microscope, we noticed that the one we induced stroke in had an elevated lipid peroxidation. Their glutathione level were reduced, and the neurons were scattered with pycnotic neurons in the cerebral cortex as well as the common amylase 1 of the hippocampus. The pretreatment with carica papaya and vitamin E mitigated oxidative and histological alterations of the neurons in both the cerebral cortex and the common amylase 1 subfield of the hippocampus. The implication is carica papaya extract protected the brain from ischemic injury. This report was published in African Journal of Medicine and Medical Sciences, Owoeye and Infection, 2018. Mr. Vice Chancellor, sir, some of the drugs that we use in treating brain conditions can also affect the brain. Sir, an example of this is phenytoin, an anticonvulsant drug we give patients that have epilepsy. The World Health Organization defined epilepsy as a recurrent, unprovoked seizure characterized by sudden excessive electrical discharge in neurons. What did we do? We studied the effects of colaviron, which is obtained from bitter cola, which have been proved over and over to have a very potent antioxidant activity. We gave another group of rats phenytoin. What the result we got was that phenytoin, this same drug we use in treating epilepsy, caused a significant increase in lipid peroxidation and hydrogen peroxide levels in the rats. It depleted the antioxidant status and significantly reduced the molecular layer of the cerebellum. And then the one we gave, vitamin E as well as the one we gave, vitamin E as a comparator, showed very different results. And the one that we gave, colaviron, completely abrogated the damages we saw by reversing it. The co-treatment with colaviron and vitamin E effectively therefore reversed the phenytoin-mediated alterations that we saw in the brain antioxidant and histomorphology when compared. The implication is that colaviron obtained from bitter cola and vitamin E may be considered as possible protective supplements for further studies in the areas of epileptic research. The report of the work was published in Toxicology Mechanism Methods, Owoeye et al. 2014. Why is it that the brain is considered more vulnerable to oxidative stress than other organs in the body? It is because there are many reasons, but three of the reasons are that the brain is only 2% of the total body weight, yet the brain consumes high amount of blood supply to the body. 2, it contains high content of polyunsaturated fatty acid, and it has a low level of antioxidant enzymes as compared with others. What are the other areas that I worked on? Or is it only the brain you have been working on? No, I study other areas. Mr. Vice Chancellor, sir, in a study conceived and led by Professor Owoeye, then of chemical pathology, now the foundation head of Department of Immunology, we studied the activity of vitamins and antioxidants activity on the effect of dicophos. an organophosphorus compound that we use commonly as pest control, either as household or agricultural pesticide. Daclofos is otherwise known as Otapiapia or Nuvan or Sniper in southwest Nigeria. And we noted that it's neurotoxic. Why? Because it impedes the acetylcholinesterase, resulting in excessive accumulation of acetylcholine in synaptic clefts. In the result that we had, having exposed the rats by inhalation for 4 hours daily for about 1 to 6 weeks, what did we find? We found that the lung was severely damaged with wide extension of the bronchial-associated lymphoid tissue, BLT, and generalized congestion of the peribronchial and interalveolar septum when we compare with the control. And then when we look at the liver showed a lot of vascular degeneration. The hepatocytes are dying. There was periportal cellular infiltration. There was vascular degeneration, and the liver cells were necrosed. But then the three groups that we treated with vitamin E, vitamin C, and red palm oil reversed all these alterations. What is the implication? The implication is that it is not when people drink Otapiapia. No. Even when they inhale it can cause histopathological changes in their lung, histopathological changes in their liver, and then it could kill them. This work was reported in the European Journal of Anatomy, Owoeye et al., 2012. Concluding remarks. Mr. Vice Chancellor, this lecture has given me the opportunity to share with you the occurrence of set of 3 related items in many topics in the brain that I saw repeatedly as I studied the human brain. Learning, understanding, and recall of learned event is easily enhanced when the learned information is linked or connected together, rather than when you learn it isolatedly. Linked or connected items consisted of related information is easy to assimilate and easier to recall during examination, during discussions, during quizzes, or during anatomical examinations. Of the 66 of such items that I listed in my book, I was able to talk to less than 1/3. I therefore dedicate this first part of my lecture to all our anatomy students of medicine, physiotherapy, physiology, pharmacy, nursing, DLLS, human nutrition and dietetics, and environmental health science. My students, the second part of my lecture highlighted some of the various sources of injury that can affect the brain and some natural substances that can ameliorate or mitigate these injuries, all aimed at protecting the brain. We may say that all organs of the body are equally important. After all animals are equal, but some are more equal than the others. But I want to say to you that some muscles may be absent in the body. There is a disease we call Poland anomaly, whereby a man may not have pectoralis muscle, a muscle of the chest wall. The muscle we use to flex the arm or to medially rotate it, and yet he will live. There's even a man that doesn't have 2 hands and doesn't have 2 legs, yet is living. But any defect or disease or injury of the brain or spinal cord will reduce the quality of life and make the victim dependent and become a burden for the family and the society. The human brain, therefore, though small, is relatively concerned with intellectual function and all the lists that I've given you earlier on. Decision making and capacity for speech and language. It is true that our experimental results were derived from laboratory rats. I agree. And I know that we cannot extrapolate results from rats directly to human beings. Yet, genetic studies have shown that there is a similarity between the genome of man and that of rats. In drug development, for example, the rat is routinely employed to demonstrate therapeutic efficacy. I expected to assess the toxicity of the drug compounds. Therefore, the takeaway I'm saying is that we are to carefully, jealously, and like a boxer, fiercely protect our brain as much as possible. For the brain is the headquarters of the command structure of the human body. So I conclude and say, as much as possible, protect your brain from all injury and insults. Recommendation. Our recommendation is that with the increase in the number of MBBS and BDS students in our university, the government will do a commensurate increase in academic, technical, and administrative staff so as to reduce the strain and you put on them. Acknowledgements. I am grateful to my institution, University of Ibadan, knowledge, and also to contribute knowledge back. This university gave me 3 degrees. Undergraduate degree, MBBS, postgraduate degree, Masters, and a PhD. So academically, I belong to the class of people that are called born and bred and butter in UI. First degree, second degree, third degree. I also want to thank the Vice Chancellor for giving me the opportunity to work in this great citadel of learning and making me part of those who impart knowledge. My immense gratitude goes to the 13th Vice Chancellor, Professor Kayode Adebowale, FAS, MNI, for the love he has for my department. For example, the renaming of our SY Anatomy Lecture Theatre to APO Desalu Lecture Theatre after the Governing Council approved it in 2022, he did. He also has vigorously pursued the acquisition of transmission electron microscope for our university and it is domiciled in the Department of Anatomy. Good news, the machine is already in the Department of Anatomy. Physically, a very good example. This is a very good example of continuity of administration. The 12th Vice Chancellor of this university, Professor Iberi Dokola, started the process. Initiated by my HOD, Professor M. Shokunbi. This 13th Vice Chancellor did not abandon it, he pursued it to a logical conclusion. I also pay tribute to my colleagues in the Department of Anatomy, academic, administrative, and technical staff. I'm greatly indebted to all that you are putting to see that our department runs. Anatomy department is unique. Sometimes I'll stay in that department till 11:45 midnight to receive the dead body of somebody. That is the sacrifice we make, but we university very much appreciate. My next gratitude is to the Professor M. Shokunbi, who was the HOD and later the 6th Provost of the College of Medicine, resuscitated the postgraduate program, initiated and pursued it that made me employed in 2001. My first academic mentor and research guide, Venerable Professor Owoeye Malomo, in neurosurgeon, I thank you. My faculty, I thank my dean, Professor Yoruke Odunola, and all my past deans. My present dean recommended me to the Vice Chancellor for this inaugural election, for which I am grateful, ma'am. I appreciate and thank all professors in our faculty, all other lecturers, administrative, our FO and his staff, and every other technical staff. God bless you all. My college, the College of Medicine, the Provost of the College of Medicine, Professor Steve Ogundiran, the Deputy Provost, Professor Diolale, for leadership, for encouraging me, and then for my department, the Secretary to the College, the FC, the College Librarian, and all the other days in the faculty. Veterinary medicine, my department with veterinary medicine. That was where I pursued part of my MSc. That was where I did my PhD, where the late Professor Omuka accepted to supervise me, and Professor Bambali Oke was the one who graduated me. My classmate, Professor James Olukayode Olokade, who were MSc class, and Professor Stephen Apayi. Also from vet medicine, Professor Tunde Otesile, who kept on giving advice and encouragement. My biochemistry connection, supervision and mentorship. Professor E.O. Farombi agreed to supervise me at the PhD level. He taught me the culture of research, ethics, work values, and all, especially how to write journals. And that if they reject your journals, what you should do to make them accept it in another journal. He has been an encourager, a peer supporter, an helper of destiny, a trainer, research collaborator, mentor, and the unique angel God placed on my path. He opened his laboratories to me and for all the work we still do. On March 8 this year, he was announced by the World Ranking of Scientists as #1 toxicologist in Nigeria and top 1% in the world. I acknowledge and thank my spiritual father who begot me into the gospel in the order of 1 Corinthians 4:15, Pastor Terry Emmanuel. December 1993, I gave my life to Christ after his preaching. I appreciate my bishop, Bishop Thomas Arenu of the Living Faith Church, and my pastors in my pastoral church, my State Pastor, Pastor Michael Olaiwaju, my assistant pastor, Pastor Ayokule, and all the other pastors. My own company in that place is called the Challengers Club. I appreciate all of you. Members of the Bachelor Resident Association, I appreciate you. The first female judge in Oyo State is Emeritus Justice B.O. Adeniji. I appreciate her for her love, for her warmness, for generosity and caring. She is the best neighbor in the world. The full gospel businessman fellowship international, FGBMA5. Let us serve him together. Why? For his banner over us is love. I acknowledge the fellowship of the Inner Circle of Friends, University of Ibadan. Quiet but deep. Founded in 1958, it's a chance that made brothers, portals, and make friends. A special mention of made of Professors Stephen Owoeye Akpagbe. I thank my classmates at the Owoeye Grammar School, my classmates at Government College. I appreciate the continued good and caring relationship of the UI College of Medicine set of 1983. Professor Tugwale Arotiba, one of us, is the Deputy Vice Chancellor Academic of this university today. And Professor Etolu Ajayi is the Director of IMAT, also a member of our set. The professional bodies, I thank my siblings, my professional bodies, Anatomical Society of Nigeria, NMA, and this who gave me a fellowship of the Anatomical Society. I thank my siblings who have always been there for me. My siblings, Mrs. Fumilayo Adeoye ní Owoeye, Mr. Bankole is not here, but is hearing us, and then the Right Reverend Francis Olariwaju Bankole, the pioneer bishop of Ekiti South Anglican Communion. The side collaborators, Professor Iyo Farombi, Professor Malomo, the list. Then the transition electoral microscope team, Mrs. Miriam Zakaria, the deputy bossa, Engineer Ajibola, Engineer Tetolu, the director of works. I thank you for all the sacrifice you've done for us. My graduate trainees, I have acknowledged you. My old teachers have acknowledged them. My postgraduate students, numerous masters and especially my PhD students. Dr. Folarin is in Rwanda, Dr. Fundo Adileye is in Lautech, Dr. Babatunde Bitoye is in Futa, Dr. Amos Murakiwe is in the United Kingdom, and they are all doing incredibly well. I appreciate all of them, and I pray that they will continue to grow to higher heights. I appreciate the management of Ibadan University Press, Mr. Olatunde Oladejo, the director, Mr. Abibulai Oladejo, the computer operator, and Mrs. Oluwatoyin Fashion, our first contact secretary. Then my children, Oluwagbemi Gan, Oluwabukola, Badegeshi Aniyan, Oluwa Tantan, Enibobo, Boye, and Keluwa. These are the ladies. I have 4 ladies in my life. One is a wife. One is wife. I have only one wife. The other are children. Finally, then the other children I have shown. Finally, I pay a tribute the youngest member of the family is the baby they are carrying. Finally, I pay a special tribute to the lady I just met in school, the Owoeye Grammar School. Through our immense support, encouragement, and prayers, she has kept the whole phone. She is my jewel of inestimable value, and my visiting support has taught me. For 32 years of great chemistry, still going. She and her children are appreciative. The Almighty God, the giver of life and all good and perfect peace, in Him we live, in Him we move, and in Him we have our being. The second Samuel, David said, "Who am I, O Lord? And where is my house that you have brought me hitherto? And before you, this is simply grace." Mr. Vice Chancellor, sir, I want to thank you for this wonderful day for your attention.
Virginia.